Greetings, chem students. We are going to discuss the entrance slip over dihybrid crosses. In mice, the ability to run normally is a dominant trait. Mice with this trait are called running mice, capital R. The recessive trait causes mice to run in circles. Mice with this trait are called waltzing mice, lowercase r. Hair color is also inherited in mice. Black hair, capital B, is dominant over brown hair, lowercase b. Your job is to determine the parent genotypes, determine the possible gametes, and then the offspring. Then you construct the dihybrid cross to solve the problem. The problem you are going to solve is crossing a heterozygous running heterozygous black mouse with a homozygous waltzing homozygous brown mouse. First thing we need to do is we need to get the parental genotypes. So we need to get the genotypes of the parents. We are going to do that by determining a heterozygous running. So heterozygous is R, little r. Heterozygous black mouse is capital B, lowercase b. And we are going to cross that with a homozygous waltzing, little r, with a homozygous brown, little b, little b. So what we will do now is we need to determine our gametes. So I am going to use what we describe as the FOIL method. So we're going to start off with big R, big B, little b. So the FOIL method, the first, capital R, capital B. Outside, capital R, lowercase b. Inside, lowercase r, capital B. And last, lowercase r, lowercase b. So when I FOILed the first one, I had 4 different gametes, okay? Now, I'm going to then FOIL my second one, which is little r, little b. FOIL first is little r, little b. That's a little funky little r there because I couldn't erase that. O is outside, so I have a little r, little b. FOI is inside, little r, little b. And finally, L is last, little r, little b. So what we have here, if you're looking at those gametes, they are all the same. They are all the same. So we don't need to do all of those. I only need to do one of them. They're going to end up being the same. So when I make my Punnett square now, I'm only going to fill up the top row of boxes. So I'm going to have a capital R, capital B, little b, capital R, little b, lowercase r, capital B, lowercase b, and finally, lowercase r, lowercase b, lowercase b. So, I have now done my Punnett square. All I need to do now is the final part here down at the bottom is to determine the phenotypic ratios. Okay? Determine the phenotypic ratios. I'm going to shrink this down just a teensy bit so that I can still see those up there. So I shrunk it down a little bit. Hopefully you can still see this on the video. But the very first one I have here, I can see, what I do is I can see, all right, the first one is a running black mouse, okay? A running black mouse. The second one here is the second one, and I said this is the first one. Right here now, I'm doing the second one. The second one is a running brown mouse. The third one, right there, is a waltzing, and that is a black mouse. And finally, my fourth one, right there, is a waltzing brown mouse. So if I'm doing my probabilities here for my phenotypic ratio, I know that I have only 4 different things. So it's going to be something out of 4. And I can see that each one of these, okay, I have 1 running black. I have 1 running brown. I have 1 waltzing black. I have 1 waltzing brown. So if I were going to convert those into percentages, what percent is 1 out of 4? 1/4 is equal to 25%, right? So each one of these are going to be 25%. Okay, so my probabilities are going to be 25% running black mice, 25% running brown mice, 25% waltzing black mice, and 25% waltzing brown mice. This has been the dihybrid cross of biology. Okay, so this is your entrance slip dihybrid cross. Have a great day.